All right.
Hi, everyone. I'm Ashley,
I'm Iris.
and this is your week 5 review session. So, we're going to go through the 4 main pathways that were talked about this week, do some practice problems that are like problem set, maybe find exam related questions that you might see, and then go through questions you guys have and whatever else.
Yep. Okay, there we go.
So, the first pathway that we're going to talk about is the survival signaling pathway, which is used for surviving, the cells for surviving. Otherwise, if this doesn't happen, they're just going to go through programmed cell death. So the first step is the survival signal will come down and bind to the RTK, which causes the dimerization, which is just when the two molecules come together to form a functional unit. And that's through a conformational shift that'll send the signal into the cell. So then the RTKs then phosphorylate each other, which is called autophosphorylation. This allows for the PI3 kinase to bind to RTK through its SH2 domain, which is just the phosphotyrosine binding domain, and that'll activate it. So then PI3 kinase will phosphorylate the PIP2 to PIP3, and that PIP3 is anchored to the intracellular side of the membrane via a lipid. And then one PIP3 serves as the binding site for protein kinase 1, which will bind to PIP3 via its pH domain. And a pH domain is just the binding site on a protein where the triphosphorylated inositol phospholipid will bind. And then the other PIP3 will serve as the binding site for AKT, which is also through a pH domain. So then PK1 will phosphorylate AKT, and that'll cause it to dissociate from PIP3 and allow PK2 to phosphorylate AKT. So that's done twice for it to become activated. And then it'll just float around in the cytosol until it finds BAD. It'll phosphorylate BAD, which then releases BCL2, which will then inhibit cell death or apoptosis. If for some reason this doesn't happen and BAD still is holding on to BCL2, then cell death will occur. And and then we have a photo of the whole entire pathway. So if you follow all the steps through, that's pretty much what's happening. A really good tip is to draw it out yourself or find the blank one and go through and label what's happening in all of them.
Do you want to go through the pathway now that they can see it, or
yeah, sure. I mean, so this is the survival signal, and these are the two receptors that it's going to bind to, and it's going to dimerize together. And then you can see the activated receptor tyrosine kinase there. They get phosphorylated with PI3 kinase. And then it goes through with the PIP3 and the two pH domains that we talked about. So PDK1 is also a pH domain that can be used. And those are going to get phosphorylated. And that middle one is the green one with AKT. And you can see that it's being phosphorylated twice. So then that causes that disassociation away from everything, and it's activated at that point. So then down here, you can see it'll go down to BAD, which is also with BCL2 right now. And it'll either cause that top one, which will have the inactivation, and there'll be BCL2 constantly bound, and it will have apoptosis, or you can do that second route, which will be the inhibition of apoptosis with BAD being inactivated and BCL2 being able to stop apoptosis.
Yep.
So then we have the JAK-STAT pathway, which her main example in class is that it's used for milk protein production and breastfeeding. So the JAK-STAT pathway is just a heterodimer, so it's going to be those two dimers again, composed of the receptor and then the receptor binding domain on it on the outside, where that signal will bind. And then you have the JAK, which is a non-receptor tyrosine kinase on the intracellular side. So it starts off with that signal, again, the RTK pathway. So it'll have that signal bind again to the receptors. Those two receptors with their JAKs on them will come together, they'll dimerize. And then what the JAKs do instead is that they cross-phosphorylate each other via their kinase domains, and then those phosphorylated JAKs will phosphorylate their specific receptors. So then this allows for a binding site for the STAT5 proteins via their SH2 domains on the bottom area of that because of the area that the JAK's phosphorylated on the receptor, it has that opening. So then one STAT5 will bind to the phosphate on the receptor, and the other will bind to the other. And then the JAKs will also phosphorylate the STAT5 proteins as well. So then this causes both of the STAT5 proteins to dissociate, and then those two come together and dimerize, and that causes them to become active. So then the STAT5 dimer will enter the nucleus with the help of important and act as a transcription factor to upregulate transcription. So then with the picture, you can see here at the beginning, the prolactin receptor is just that breastfeeding protein that I was talking about that's going to signal for production of the milk for breastfeeding. So that'll come through. That'll be the signal. It'll have those two, the JAK1 and the JAK2, on the receptors come together. And then you can see they're phosphorylating each other with those red arrows. And then once that's been done, they're going to phosphorylate their own specific receptor, so on each side instead of the other ones.
Oh, sorry.
That's fine.
Wait. How do I go back? Okay. Got it.
And then you can see that once the receptors have been phosphorylated and all of that, the stats will come and bind, which is that orange piece down there. And the two JAKs will then phosphorylate each of those. That causes them to dissociate and then dimerize together. So then you have right there your active STAT5 dimer, and that comes down and acts into the nucleus through important. And then you get transcription of milk protein. And there's a funny meme, if anyone finds it funny, but anyway.
Okay.
And then we have the RTK RAS MAP pathway. So this can be primarily used in like cell division, stuff like that, cell differentiation. So most of these will start with the RTK pathway again. So that signal will bind on the outside of the extracellular portion. It'll bind to RTK, it'll dimerize, so the two things will come together again. And then the RTKs will phosphorylate each other at the domains. And then we have this adapter protein called GRAB2, which will then bind to the active RTK through the SH2 domain. And then there's that RAS activating protein, which may be referred to as SOS. And that specific one has the PXXP motif, which binds to GRAB2 through its SH3 domain. So the difference between the SH2 domain is just a specific or a non-specific phosphotyrosine binding domain, but that SH3 domain is going to be on GRAB2, and it's going to specifically bind to a PXXP motif instead. So, and you find that on the RAS activating protein. So then the RAS activating protein will cause GDP to dissociate from RAS, which is a GTPase, and that'll allow GTP to diffuse into RAS. So once that happens and GTP is bound to RAS, it becomes active. So then RAS slides along the inside of the cell membrane, trying to find MAPK3, and once it does, it somehow activates it. It doesn't do this through phosphorylation though, like Laura said in class. It's not really known well how it does it, but it's definitely not through phosphorylation. So just if you have a question like that, kind of keep it general, just it does it somehow, but it'll activate it because MAPK3 is not a kinase. So active MAPK3 then will phosphorylate MAPK2, and that'll cause MAPK2 to become active, and it'll keep going with that cascade. So then MAPK2 will phosphorylate MAPK, causing MAPK to become active. And then once that's finally done and all of that's active, MAPK will activate downstream effectors such as a protein or a transcription regulator, leading to cell growth and cell division. And then to kind of shut everything off, eventually RAS is going to be deactivated by GAP, and that will then bind to RAS, and it'll accelerate the hydrolysis of GTP to GDP, and that'll inactivate it because, as we know, to activate it, it has to go from GDP to GTP, so that eventually shuts it off. Yeah. So then you can see that here, we have that RTK whole beginning part with the signal molecule and everything. And then we've got that adapter protein, which is going to be your GRAB2 and the RAS activating protein of SOS. And that's, you can see, comes up and it'll remove the GDP, bring in the GTP. Then we got that RAS protein activated, and that'll go through. And then you can see the whole MAPK cascade go through with the activated RAS protein activating MAPK3, it'll go from ADP to ADP and can you continue all the way down to just MAPK. And then you can see there's multiple different options on what can happen after that last one's activated.
Yep.
Yeah.
Before we move on to GPCR, does anyone have any questions about those other pathways that we talked about? Okay, so we can move on to GPCR pathway. So GPCRs are made of 7 transmembrane receptors, and the intracellular portion is bound to a G protein that has 3 subunits. They're called G alpha, G beta, and G gamma. And the G alpha subunit is a GTPase, which means it can hydrolyze GTP to GDP. And so in its inactive state, GPCR is bound to the G protein and G alpha is bound to GDP. Once a signal binds, that signal is transmitted across that 7 transmembrane receptor into the intracellular portion, and that causes a conformational shift in G alpha, which causes it to it causes the GDP to dissociate from the binding pocket, and then GTP can diffuse in, and that activates G alpha. And so that's how the GPCR can act as a GEF to G alpha, because it causes G alpha to let go of its GDP, and that allows the GTP to dissociate in, and then G alpha can be active. So when G alpha becomes active, it dissociates from the beta gamma complex, and then it can bind to effectors or start its own signaling. And then the beta gamma complex also has its own signaling that's separate from G alpha. So they kind of do their own thing. And then the way that the pathway is shut off is when an RGS binds to G alpha. So it acts as a gap and it accelerates G alpha's GTPase activity. So it hydrolyzes, or it helps G alpha hydrolyze GTP to GDP, and then G alpha is inactive. So then once G alpha is inactive, it will come back together with the beta gamma, and then the whole GPCR with the G protein is inactive. So here's the diagram of that. The receptor will bind to the outside portion of the GPCR. The signal will travel through the transmembrane part of the receptor, and it reaches G alpha. It causes a conformational shift in G alpha, and the GDP that's in the binding pocket will dissociate, and then that allows GTP to, or I guess that's over here, GTP can diffuse in, and then G alpha becomes active. It binds to effector proteins and does its own signaling over here. And this beta gamma complex, which has a lipid anchor right here, can also bind to effector proteins and do different signaling. And then it will signal for as long as, like, it'll signal until an RGS comes in, and that helps G alpha hydrolyze GTP to GDP, and then once it's in a GDP bound state, it's inactive. So the whole complex comes together. If the signal is still bound, once it comes back together, then the whole process will repeat itself. If not, it'll just kind of be in this complex until another signal binds.
All right. Before we get into any like questions, does anybody have any questions about anything? No. Okay. So you can like answer in the chat or shout it out, whatever you want to do. But here's the first question. So which of the following would allow the cells to survive with these mutations? So this goes back to the survival signal pathway. So go through and say which one would allow for the cells to survive and which one won't, and give a reason why. Yeah. This is very similar to the one you did in class today. So
yeah, we'll see who is paying attention.
yeah.
And once you have an answer, as Ashley said, shout it out or put it in the chat.
Yeah, two. Oh, it's a multiple choice, so you can do multiples, but yes, 2 is correct. And 2 and 4 is the full correct answer. Does anybody want to say why that would be the right one? Why those 2 would allow for constant cell survival?
Do you want to just explain?
Yeah, sure. So for the first one, that would allow for cell survival, which is the gain of function AKT. Basically, if there's a gain of function AKT, it's always going to be on. So AKT will always be able to phosphorylate bad, and that'll always release BCL2 and inhibit apoptosis. And then for 4, having a loss in the mutation of PTEN, which is the phosphatase that'll take off one of the phosphates in PIP3, that's going to always allow for cell survival, and that's because PTEN will be able to phosphorylate PIP3 back to PIP2, and that causes no downstream effect. So if it cannot phosphorylate PIP3, it'll continue and programmed cell death will not occur. So yeah, do you want an explanation on why the other ones would cause cell death? Anybody? If not, we can just move on. But
Yeah, I think it would be good to explain.
okay. So the other ones would cause cell death constantly. So if you have the loss of function mutation in PI3 kinase, that's going to cause cell death because it's not going to be able to phosphorylate PIP2 to PIP3 and have that whole cycle of PIP3 being able to have the PK1 and all that continue on. So it's just going to stop there. And then having a bad protein that can't be phosphorylated but can still bind to BCL2. So basically with that one, basically bad's just going to still be able to bind to BCL2 regardless if it's going to be phosphorylated or not. So it'll just stay bound to BCL2, which we know will then just cause cell death. Yeah.
All right. Does
Anybody
anyone have questions?
Does that make sense? can give a little yes in the chat if it does anything. Perfect. Awesome. Thank you, Jillian.
All right. In that case, we'll move on to the next question. All right, which of the following mechanisms would work to shut down signal transduction in the RTK RAS MAPK pathway? So for each one, give a yes or no answer and explain why. Yeah, that's right for number one. Mutating the kinase domain of RTK would shut down the pathway because the kinase domain of RTK gives the receptor the ability to autophosphorylate. So if it's mutated, there will be no autophosphorylation, and then downstream proteins won't be able to bind and become activated. And so that would like inhibit the entire signaling pathway.
What about 2 and 3, everyone?
Is anyone confused about what number 2 means? Okay, so one person says yes for number 2, one person says no. Do one of you want to explain why? Fight it out.
Yeah.
Okay, so in this case, the extracellular portion of the receptor would be inside the cell. So no signals would be able to bind to the receptor because the RTK is endocytosed into the cell, and the cell wouldn't be able to recognize if there's a signal. The signal wouldn't be able to bind, and then the pathway wouldn't be able to become activated, and the signal wouldn't be transduced. So that one would work to shut down the signal transduction pathway. And then any ideas for number 3? Okay, so number 3 says mutate RAS such that it cannot hydrolyze GTP. So if RAS can't go from a GTP to GDP bound state, it means it can't become inactive, because when it's bound to GTP, it's in its active state. So if RAS can't hydrolyze GTP, it will always be active, and it will always be transmitting the signal by activating downstream components of the signal transduction pathway. So if it keeps activating the pathway doesn't shut off. And so it would be overactive, and this mutation wouldn't work to shut down the signal transduction, because it'll just, like, this mutation would cause it to be overactive. Does that make sense? Does anyone have questions on that? Okay. All right, next question. Gain of function mutations. So what would be the effect on GPCR signaling if each of the following had a gain of function mutation? So answer the question for if the GPCR had a gain of function mutation and G alpha and RGS, like separately. Any ideas for gain of function mutation in GPCR? Oops, sorry. Oh, whoops. I didn't mean to change the slide. Okay, so if the GPCR had a gain of function mutation, it would mean the pathway would always be on, because it would constantly be transducing a signal, even if the signal wasn't bound to GPCR. And then that means that G alpha would always be in a GTP bound state. And when G alpha is in a GTP bound state, it's transducing a signal because it's active. So then a gain of function mutation in GPCR would cause an overactive signaling pathway. And in G alpha, gain of function mutation would mean that the pathway would also always be on for pretty much the same reason, because G alpha wouldn't need the GPCR to become active. If it has a gain of function mutation, it means that it would always be active. And then that relates to what I was saying before. If G alpha is always active, then it will always be participating in transducing the signal. And then that would cause an overactive pathway. And gain of function in RGS. So RGS helps G alpha hydrolyze GTP to GDP. If there's a gain of function in RGS, then it would be constantly doing that. So it would cause G alpha to be in an inactive state all the time, pretty much. And then that would shut down the pathway, because when G alpha is inactive, it can't transduce a signal. So no signaling would occur if there were to be a gain of function mutation in RGS. And I would go through each of these for loss of function mutations too, and try to quiz yourself to see if you know what would happen to the pathway if there was a loss of function mutation in each component of the GPCR pathway. Oh, any questions before we move on? Okay, so this is another question about lipid anchors. The question is, what's the purpose of having a lipid that anchors the proteins to the inner surface of the cell membrane? And how is this localization versus having the proteins be freely diffusing in the cytosol advantageous to signal transduction? Does anyone have any ideas for this one? Someone take a guess, or
Think about how stuff is transmitted like extracellular to intracellular. So if that helps at all, where it would need to be right on the intercellular inner surface of the cell if the extracellular is going to transmit it directly through instead of in the middle of the cell or
okay, so for this one, think about how proteins with lipid anchors interact with other molecules in the membrane. So if they were floating around in the cytosol, it would be harder for them to interact with the proteins that they need to, or molecules that they need to interact with. So it's more efficient for them to have a lipid anchor that allows them to diffuse laterally across the cell membrane. And this is advantageous for signal transduction because it makes it easier for the components to, like the components needed for signal transduction that need to be close to one another. It makes it easier for that to happen. And it also allows the signal to be transduced faster.
anybody have any questions about anything? I believe that's the last question for practice problems. So if not, we can stop the recording.
All right. In that case, we'll stop the recording.